Thank you. It's my pleasure to introduce the next speaker, Dr. Monkia Jevsnik, with the title of Review of Diagnostics of Non-SARS Human Coronavirus in Slovenia. What have we learned? Just to give you a little bit of background on this next lecture, Monkia comes about a little bit more than 10 years ago to our laboratory. And because we have an opportunity to use electron microscopy for diagnosis of gastrointestinal viral infections, we actually look for the same patient, the symptoms of gastrointestinal infection and the same time of the respiratory infections. And our colleagues from the department, they sometimes find coronaviruses in the stool by electron microscopy. And Monkia gave us idea to maybe would be interesting to look at those kind of patients who have both systems involved in the disease. So they have at the same time gastroenteritis and respiratory symptoms. So this was actually the beginning of our coronavirus research, and as I told you in the morning, it was quite frustrating at the beginning because when we sent out our manuscript, the reviewer said our data and scientific works is sound, but it's clinically unimportant, so we will not publish it. So we need about 10 years now, and Monkia would probably told you what we learned in this last 10 years of work, which begins like I told you now. So please, Monkia.
Good afternoon, everyone. So thank you, Professor Petrovec, for so nice introduction. So our beginnings and our journey started in June 2007 when we started with a retrospective study. Before this year, human coronaviruses were not included in our respiratory diagnostic. After 1 year, we also decided, because of the very interesting nature of the human coronaviruses, that we started with prospective study from October 2009. So prospective study lasted 2 years and included children under the 6 years of age, also with both symptomatic. After 1 year lasting of this study, we included human coronaviruses in routine respiratory diagnostic. And we totally changed our diagnostic from direct immunofluorescence assays to the in-house real-time PCR assay. So we also included a lot of other respiratory viruses that, as you can see here in this scheme. From 2018, we began switch our diagnostic from in-house to the IVD method, house diagnostic respiratory panel. And also from 2021, we included SARS-CoV-2 in our routine diagnostic. So I will show you our results from previous studies, and we done a lot of different aspects of human coronaviruses. So before the 2007, we have no idea about circulation of human coronaviruses in Slovenia. So we don't have any insights of prevalence and what they cause in Slovenian children. So interesting was that we get with both studies really similar respiratory seasonal distribution of these viruses. with the maximum peak in the February. And the prevalence with retrospective study was 6%, and in prospective study, so from June 2007 to, October 2009 to September 2011 was 8.1%. And all these distributions you can see on the both figures, so on the left and on the right side. We also detected all 4 species of coronaviruses in all 3 years, with most prevalent HKU1. And here in this figure, you can see the distribution of all respiratory viruses in very long period. So from between January 2012 to July 2023, on the left axis, you can see respiratory virus prevalence, and on the right side, the total number of all viruses and all samples that were sent in our laboratory to for routine diagnostic for respiratory viruses. You can see that it's very crowded here, and that every virus has his own position in the year. And on the figure below represents the seasonal coronaviruses with the light green, and in the dark green line you can see the SARS-CoV-2. So before the epidemic, we can see that seasonal pattern is mostly repeated every year, and with the maximum peak in January or in February. But you can see here, in 1 year before the SARS-CoV-2 epidemic, we can see that we have change in the pattern, and probably because we had this year very high and strong flu A and B season, and also RSV. These are also the data that we saw that we detected all 4 species of coronaviruses in this period, but here we have no results, because these are the results from the samples that were tested with film array. So I hope that in the next few years we will typing all those viruses. I hope so, and it will be interesting to see the results of maybe a bigger study. In prospective study was more clinically based. So we included, as I already said, children under the 6 years old, and we included children with 3 clinical presentation. So children with acute bronchiolitis, acute gastroenteritis, and febrile seizures. Also, we also included children healthy control group without respiratory and gastrointestinal symptoms. Nasopharyngeal swabs and stool samples were taken from all children, and blood samples additional from the symptomatic children, and also those children were also follow up 40 days after the initial sampling. We can see here that clinical insights is very complex and difficult to explain, and more probably it's less common and important in children with acute bronchiolitis and acute gastroenteritis, but more likely in children with febrile seizures. Why? Because they has a lot of positive results in these children, and also the highest viral load that is not represented here. On the other hand, here on this figure, you can see that a lot of children, this is a very high percent of them, so 95% has co-infection with other respiratory viruses, so children with acute bronchiolitis. And on the other hand, here in children with gastroenteritis, in stool samples, we also had a lot of co-infections with other gastroenteric viruses. So it's a little bit complex to explain. Also because we have here in children with gastroenteritis, they had first of all more virus detected in nasopharyngeal swabs than in stool samples, and on the other hand has less co-infections than children with acute bronchiolitis. This is a little bit controversial. Okay. But if we look more closely these children, so we saw that most of the children had also upper respiratory tract infection. And this percent is rise up to 80% if the coronaviruses were simultaneously detected in both samples, so in nasopharyngeals and in stool samples. So most of the children had also acute respiratory tract infection. But when we are looking to follow-up samples, so we didn't find any differences in the prevalence from baseline to follow-up. But interesting is the fact that most of the coronaviruses that we detected in the follow-up samples were newly detected. So only around 30% was those that were detected at a baseline and also at follow-up. Here in this figure on the right side, we can see the CT values detected for those viruses that were species of coronaviruses that were detected in both sampling. And as you can see that is, as we expected, the CT value at follow-up higher, but only in NL63 is surprisingly the lower. But we have very small sample with such both sampling, so in the both samples. So transmissible insights for coronaviruses we saw very, very early. So in 2012, we in some the study were included 55 patients hospitalized in intensive care unit and 46 employees, and we saw that the coronaviruses were the most common and most transmissible viruses between patients and employees. So you can see this in these blue dots on the figure. And the last one that we also we were also looking for is zoonotic potential. So very close relation between OC43 and bovine coronaviruses in the same time period, but these are the results from Sanger sequencing. So maybe this is also the opportunity to do. Now we have more available methods for this sequencing. And in the end, I would like to conclude that in Slovenia, all 4 human coronaviruses are frequently detected as human pathogens, and were often co-detected with all respiratory viruses, especially in young children. That's why has minor role in children with acute bronchiolitis, acute gastroenteritis, and maybe more likely in children with febrile seizures. We also saw that the long persistent in respiratory and stool samples. And maybe I will conclude with some thought that researching coronavirus is like researching the oceans. So you never know when you'll find new species, and good collaboration with other, with researchers and clinics here in Slovenia is, I think that it's needed, and it's very important to get the right results, and also to do really good diagnostic. Thank you for your attention.
Any questions?
Yeah.
It's a beautiful data set. I'm curious about the interaction in the epidemiology of the seasonal coronas and flu or RSV, but also SARS-CoV-2, where it didn't look like they were interacting. Do you have any explanations or hypotheses?
Maybe, yeah. I think that is because seasonal flu A and B is, I think that is more pathogenic than coronavirus. You know, if some virus come into the population that is maybe stronger than others, so we saw every year that some other viruses go down and then remove and take a place for the more pathogen virus. But we didn't find any co-infection with coronavirus, with flu A and B, and also RSV. The most of the co-infections were with rhinoviruses, adenoviruses, and BOCA.
Any other question?
Yeah.
Okay.
I wonder if you did a repeat of your experiment with a fully adult population, what do you hypothesize would be the differences?
Fully adult. Yeah. I think that it will be differences between them because we are a laboratory for diagnostic of respiratory tract infections, so respiratory viruses. And before the SARS epidemic, so the most of the samples in our lab were from pediatric patients. After the SARS epidemic, then we can have also more adults. So that's why also we decided to include in our study only children, because we have samples from them. The population for coronaviruses is not, so pediatric population is not really good to see epidemiology of coronaviruses.
In adults, yeah.
Yeah, yeah.
The only adults which were tested actually systematically was the one with the prospective study in the intensive care, actually. Yeah. And this was for us a big surprise because we tested it like without indication. So the prospective study lasting 2 months, include all patients being hospitalized in the intensive care unit and all employees with regard of their symptoms. So it was just, let's see what they have. And we were very surprised, and for us should be a warning sign. It was briefly mentioned, but to explain you, what we found actually was that the coronavirus infection in the intensive care unit start with the infected patient, come to the end of the employees, and then just go between both groups. So all of the 2 months, coronaviruses are spreading between the patients hospitalized and the employees being there. And at that time, it was like, how to say, not normal to say people with some sneezing, just stay home or whatever. And what was for me most interesting when we're looking back, this was in 2009, what we found actually that all other viruses stay on the side of the population. So flu and RSV being present in employee was not spread to the patient. But coronavirus is circling between both. So this should be quite strong message that with the normal procedures, with the mask used in the intensive care units at that time, is preventing some viruses, but not coronaviruses. But we didn't get this message so as we should, because this could help us when we instituted by the coming of SARS, how infectious actually the coronaviruses, even the old ones, was at that time. But really, when Monica did a PhD, all the results we gather, the clinical part, it doesn't look like it's really important for children. I remember at some meeting in the United States where there are many clinicians, when one, I think it was a pediatrician, stand up and say, "You're a microbiologist. It is nice that you can show us like 12 viruses, but please could you explain me what it means we define the coronavirus, what clinical importance they have?" And I remember one of the lectures from Fred Hutchinson Cancer Center. They say we exclude coronaviruses from our panel because we found that they have no clinical impact at all on the outcome on our patient. So I think it's still much to learn. And as Monica said, for me personally, it was also very interesting, this high rate of co-infections. And we still get like questions from clinicians. Could you explain us, if we get 4 viruses, how should we decide who is the culprit really in the etiology of the clinical pictures we see right now? So I think another approaches are ahead to decide if there are any or any circumstance that they are clinically important in advance as it was questions.